Why did Rudolf A. Marcus, United States, win the Nobel Prize for Chemistry?
For his contributions to the theory of electron transfer reactions in chemical systems. Marcus received the prize for his work studying electron transfer reactions and developing the theory surrounding this elementary chemical process. The transfer of an electron between two molecules is a ubiquitous process in chemistry, and it is important to a wide range of chemical phenomenon, ranging from the conduction properties of materials to chemical synthesis, to the capture of light by plants for the purpose of harvesting energy. Marcus completed much of the work surrounding the theory of electron transfer in the 1950s and 1960s. And he was able to explain the greatly varying rates of electron transfer observed in different chemical systems. Some aspects of his theories were difficult to confirm experimentally, and were not proven until the 1980s. Which contributed to why it took so long for him to receive the Nobel Prize for his work.
Why did Alan J. Heeger, United States, Alan G. McDermid, United States/New Zealand, and Hideki Shirakawa, Japan, win the Nobel Prize for Chemistry?
For their discovery and development of conductive polymers. This prize was awarded for the discovery of plastics/polymers that can, under certain circumstances, be made conductive. This was quite a surprising result, since nobody ever expected plastics to be conductive materials. These plastics/polymers consist of chains of conjugated carbon-carbon double bonds. In other words, carbon-carbon double bonds that alternate with carbon-carbon single bonds. These polymers were also doped, which means that electrons were either removed or added artificially by oxidation or reduction.
What are some industrial processes that make use of catalysts?
The chemical industry uses catalysts in a staggering array of chemical reactions that make products that we all use every day. The production of ammonia, NH3, from nitrogen, N2, and hydrogen, H2. Known as the Haber-Bosch process, uses iron or ruthenium catalysts. This reaction is used to generate about 500 million tons of fertilizer per year. Most of the world's plastic is made by one form of catalysis or another. And the refining of crude oil into gasoline and other fuels is also possible because of catalysts. Catalysts are also used in food processing and production. Margarine is made by reacting fats with hydrogen gas in the presence of a nickel catalyst.
How can I extract iron from oatmeal or breakfast cereal?
Chemistry principles encountered in this experiment: magnetism, food chemistry / nutrients extraction, materials you'll need. Oatmeal experiment iron fortified instant oatmeal packet. Check the label to ensure iron content. Magnet. It's easiest to see the iron if you can find a magnet that is coated or painted white or another light color. Plastic bag or bowl. Bre fast ceril experiment magnet for this experiment. You will want a magnet you can use to stir a liquid. It's easiest to see the iron if you can find a magnet that is coated or painted white or another light color. Plastic bag, water, large glass jar or beaker. The procedure: oatmeal experiment. Open the oatmeal packet and empty it into the plastic bag or bowl. Stir the oatmeal with the magnet. You should see small amounts of gray or brown metal collect on the outside of the magnet. This is iron. Iron is commonly added as a mineral supplement to breakfast cereals and other foods. Now you can see that the iron that goes into your diet is the same element that you find in objects made of iron metal, just in much smaller quantities and pieces. Recall that iron is attracted to magnets due to the fact that it is a ferromagnetic material. See atoms and molecules. Bre fast ceril experiment. Pour 1 or 2 cups of breakfast cereal into a plastic bag. Crush the cereal inside the bag using your hands. Pour about 1 liter of water into the jar or beaker, and add the crushed cereal from the bag to the water. The water will help to extract the iron from the crushed cereal. Whereas the iron bits were looser in the dry oatmeal sample, they tend to be stuck within pieces of cereal. Which is why you need to mechanically crush the cereal and use the water to help extract it. The magnetic interaction wouldn't be strong enough to pull the iron out of the cereal on its own. In a chemistry lab, acids would often be used to help extract metals from a sample. But will work fine for our purposes here. Use the magnet to stir the crushed cereal for about 15 minutes. When you remove the magnet from the water, you should see iron filings collected on the magnet.
How long ago was the light we see from stars emitted?
To figure out how long ago the light we see from stars was emitted, we have to know roughly how far away the star is and use the known speed of light, approximately 3 × 108 meters per second. The Sun is roughly 150 million kilometers from the Earth, from which we can calculate that sunlight reaching the Earth left the Sun roughly 8 minutes ago. The distance to the next nearest star is much farther, roughly 410 × 1011 kilometers from Earth. This translates into a time of over 4 years between when light leaves this star and when it reaches telescopes on Earth. Keep in mind that this is the next nearest star, so all others are even farther away. This also means that, if we see a star explode, it really happened many years ago.
What is a monomer?
a group of atoms that forms the repeating unit in a polymer.
What is group theory and how is it useful in chemistry?
Group theory, in the chemistry sense, involves making use of the symmetry of a molecule to better understand its physical properties. By examining the symmetry properties of a molecule, it can be placed into a point group that describes the symmetry operations. Rotations of reflections, that characterize the molecule's symmetry. Group theory allows chemists to understand an impressive number of molecular properties, including the spacing of energy levels of the orbitals in a molecule, the symmetry of the molecular orbitals, the types of transitions that can occur, like vibrational and electronic excitations, and what the delocalized vibrational motions of a molecule look like. It is very impressive that symmetry can tell us so much about molecules without the need for any complex calculations.
What is resonance?
Resonance is a way that chemists represent delocalized electronic structure. Let's take that statement apart to understand what it means. Delocalized means that an electron, or a pair of electrons, is not located entirely around a single atom or bond. Take a look at the two structures of nitrogen dioxide, NO2, on the following page. The negative charge is located on one oxygen atom in one resonance structure, but can be found on the other oxygen in the second resonance structure. Notice that we said electronic structure and haven't said anything about atoms moving here. That's because they don't. Resonance only deals with electrons, and the atoms are located in the same arrangement in every resonance contributor. This is important, and makes sense if you remember that the electrons aren't moving from one resonance structure to another. These structures are needed because the actual molecule is more complicated than our simple drawing system can represent.
What is a bimolecular substitution reaction?
From the last answer, you can probably guess that a bimolecular substitution reaction has two bi molecules, molecular, in the transition state. This means that the slowest step involves two molecules interacting. In the previous example, there was only one. Let's look at a similar substitution reaction, but instead of T-buletin Cl, we'll react hydroxide with MeCl. Here the nucleophile, O, directly displaces the leaving group Cl, without forming a carbocation intermediate. This is because the methyl cation CH3 stable than the tert-butyl carbocation formed in the previous question.
What is a decant?
To pour off a liquid from a solid sediment.
What are pigments?
Pigments are molecules that selectively absorb particular ranges of wavelengths of light. The remaining reflected wavelengths are the color that you perceive the pigment to be. So pigments can only subtract wavelengths of light from the spectrum. But they cannot generate their own light. For example, a blue pigment absorbs red and green light, but reflects every other color. Alternatively, the blue pigment might only absorb orange light, which is the color complement of blue.
What is calorimetry and what is it used for?
Calorimetry involves measuring the amount of energy released during a chemical reaction in the form of heat. This is accomplished using a device called a calorimeter. A calorimeter is a container inside which a chemical reaction can be carried out while thermally insulated from the surroundings. This can even be something as simple as a sealed styrofoam, or coffee cup with a thermometer inserted through the lid, though more sophisticated devices certainly do exist. The temperature change inside the calorimeter can be monitored to determine the amount of energy given off during the chemical reaction, which can then be used to determine the enthalpy change associated with the reaction.
How does ethanol work as a fuel?
Ethanol works as a fuel in much the same way as gasoline. It is burned in a combustion reaction to release energy. In automobiles, ethanol is usually mixed with gasoline.
What is an ultraviolet?
Electromagnetic radiation with wavelengths of 10 nanometers to 400 millimeters. Just shorter than the visible spectrum, but not as short as X-rays.
What is a polar?
A compound in which the charge distribution leads to an overall dipole moment.
What is a phase?
A state of matter, e.g., solid, liquid, or gas.
How can I make jello, that glows under a black light?
Chemistry principles encountered in this experiment: fluorescence gels, cross-linking materials you'll need. Jello, R, or gelatin powder, 1 cup of tonic water, 1 cup of water, stovetop or microwave large bowl pot. For heating on stovetop, a black light source. See Physical and Theoretical Chemistry to review how black lights work. The procedure: heat 1 cup of water to a boil. Mix the Jello, R, and the hot water into the bowl, and stir the powder in until it dissolves. The hot water helps the gelatin to dissolve and disperse evenly throughout the solution. Gelatin is a form of collagen, see Chemistry in the Kitchen, and when it cools, the Jello, R, will reform cross-links between the collagen strands, which is what traps the water inside to create a gel. You'll recall that a gel is solid material that consists of a bonded network of long strand molecules that contain significant amounts of molecules that would otherwise behave as a liquid trapped within the solid network. Add in 1 cup of tonic water, stir the solution well, and then place it in the refrigerator for about 4 hours. The tonic water contains a molecule called quinine that will fluoresce a bright blue color when excited with the appropriate wavelengths of light, which can be provided by the black light. Recall that fluorescence takes place when a molecule absorbs light at one wavelength, relaxes to release a fraction of that energy, and then emits a photon at a longer wavelength, lower energy, than that which was absorbed. Black lights provide light that is at slightly shorter wavelengths, higher energies, than light in the visible spectrum. So it can often excite molecules that will fluoresce in the visible region of the spectrum. When the Jello, R, is finished hardening, take a look at it under the black light. It should glow blue. This is due to the fluorescence of the quinine from the tonic water. This blue glowing color comes from the fluorescence of the quinine molecules. So it will not be affected significantly by the flavor or color of Jello, R, that you decided to use.
What is the smallest quantity of lead that can be detected in drinking water?
Lead is something we definitely don't want in our drinking water. The maximum contaminant level goal set by the U.S. Environmental Protection Agency as of 2011, is 0, indicating that there is no amount of safe exposure to lead in drinking water. The EPA's maximum contaminant level, which is based on more practical considerations, is 15 parts per billion.
Is caramelization the same thing as the Maillard reaction?
Caramelization is the breakdown of sugar molecules with heat. Pyrolysis, while the Maillard reaction requires amino acids, proteins. Caramelization, like the Maillard reaction, is a term for hundreds of different chemical reactions taking place at the same time.
How many types of nucleotides are found in humans?
Nucleotides are named for the nitrogenous, nitrogen-containing base incorporated into the nucleotide, and there are only 5 types of bases used: adenine, guanine, cytosine, all found in both DNA and RNA, and thymine, DNA only, or uracil, RNA only. The corresponding nucleosides are called deoxyadenosine, DNA/adenosine, RNA, deoxyguanosine. DNA/guanosine, RNA, deoxycytidine, DNA/cytidine, RNA, deoxythymidine, DNA, and uridine, RNA. All of the genetic information in our body is stored in our DNA using sequences of only these 4 nucleotides. Imagine trying to describe how to make and operate an entire machine using only 4 letters. That's just what our genetic information does.
Why did Sir William Ramsay, United Kingdom, win the Nobel Prize for Chemistry?
For the discovery of the inert gaseous elements in air, and his determination of their place in the periodic system. Lord Rayleigh noted in a lecture that he measured a difference in the density of nitrogen made synthetically in the lab, and the same substance isolated from air. Ramsay followed up on this observation, quickly isolating argon for the first time. He went on to discover neon, krypton, and xenon.
What are auxetic materials?
Auxetic materials have a unique property: when they are stretched, they actually become thicker in the directions perpendicular to the applied force. Think about this in comparison to anything else you stretch. It is really quite a strange phenomenon. This occurs as a result of hinged arrangements within the material that flex apart when a force is applied to stretch it.
What is enthalpy?
Enthalpy is a measure of the energy that something contains, and it's defined as the total heat content of a system. In terms of chemical reactions, we are most often interested in the change in enthalpy, denoted H, associated with the reaction. The H for a reaction is defined as the enthalpy of the products minus the enthalpy of the reactants. And this is typically measured via changes in temperature that take place during the reaction.
Why does my chocolate turn white when I wait too long to eat it?
Chocolatiers refer to this as a bloomed chocolate. which is a wonderfully obscure and appealing way of describing the white stuff that forms on old Halloween candy. There are two processes that could be happening here, either sugar bloom or fat bloom. Sugar bloom is the candy world's way of saying sugar crystallization. If your candy is exposed to moisture, sugar molecules dissolve out of the fat in the chocolate, and once that moisture evaporates, the separated sugars have a chance to crystallize. If your candy has stayed dry but underwent a quick temperature change or was stored warm, it's probably fat from the cocoa butter that has separated from the chocolate. In either case, it is usually still fine to eat.
What is a sugar?
Sugars are part of a class of biomolecules called carbohydrates that are made up of carbon, hydrogen, and oxygen atoms. The simplest sugars are called monosaccharides. But, like amino acids, they can polymerize to form disaccharides and oligosaccharides. Sugars can usually exist in either a ring form or an open chain form, as shown in the picture below. The sugar in this picture is called glucose, which is the sugar most commonly used for energy in the human body.
What is the native state of a protein?
Since there are so many atoms in a protein, there exist a very large number of possible conformations into which a protein can fold. The native state is the conformation in which the protein exists in its natural biological environment. This is most often the lowest energy conformation the protein can adopt. A protein in its native state is able to carry out its biological function, while a protein that is unable to reach its native state often will be unable to do so. If a protein is taken out of a cell, for example, the pH or other factors related to its environment may cause it to adopt a conformation other than its native state.
How do nuclear power reactors work?
A nuclear power reactor works by generating heat from a controlled fission reaction, which then generates steam used to drive turbines to generate electricity. The fuel for the nuclear reactor is typically uranium-235 or plutonium-239.
How do herbal medicines differ from modern medicine?
Modern pharmaceutical medicines usually contain only one active ingredient, or a few at most. The rest of the ingredients in a pill are there to aid in its delivery in one way or another. Herbal medicines, because they are made from plants that were once living, can contain many more chemicals, though only one may be the active ingredient in this case as well.
Do electrons, protons, and neutrons all have the same mass?
Protons, neutrons, and electrons each have different masses. Electrons are, by far, the lightest of the three. With a mass of only about 1/2000 th that of a proton or neutron. Protons and neutrons have similar masses, with that of the neutron being just slightly higher than that of the proton. The masses of the three particles in kilograms are: electron mass 9.1094 x 10 to 31 kilograms, proton mass 1.6726 x 10 to 27 kilograms, neutron mass 1749 x 10 to 27 kilograms.
What are some applications of functionally graded materials?
Recall from above that a functionally graded material is one that varies in one or more properties throughout its dimensions. These constitute a relatively young class of materials with promising applications in a variety of areas. For example, the living tissues in your body, including your bones, are classified as natural, functionally graded materials, so if scientists want to develop materials capable of replacing these, they are looking to develop a functionally graded material. They are also useful in aerospace applications, where materials that can withstand a large thermal, temperature, gradient are needed. Functionally graded materials are commonly found in energy conversion devices and have also been used in gas turbine engines. They can also be good at preventing the propagation of cracks through the volume of a material, which makes them promising candidates for defense applications like developing bullet-resistant materials to create armors for humans and vehicles.
What fraction of plastics used in the United States are recycled?
In 2008, about 6.5% of the plastic waste generated was recycled. Roughly another 7.7% was burned to generate energy, while the majority of the remaining waste went into landfills. It is interesting to note that, as plastic production has continued to increase, the fraction of plastics being recycled has decreased. In some cases, this may simply be due to the increasing volume of plastic products or to the fact that it is not easy to build a profitable business around recycling plastics.
What is an enzyme?
An enzyme is a protein whose function is to serve as a catalyst for a chemical reaction within a biological system. Some functions that enzymes perform are the synthesis of proteins and other biomolecules, digestion of fats and other molecules, and sometimes they are even put to use in industrial applications outside of their natural biological environment.
Why did Francis William Aston, United Kingdom, win the Nobel Prize for Chemistry?
For his discovery, by means of his mass spectrograph, of isotopes in a large number of non-radioactive elements. And for his enunciation of the whole number rule. Following the prize in 1921 for Soddy, the Nobel Committee again recognized the importance of isotopes by selecting Aston for the prize. Aston built the first instrument capable of separating individual isotopes of a given element, which he used to identify over 200 elemental isotopes. This work led him to conclude that all isotopes of all elements have whole number masses. If one defines the major isotope of oxygen to be 16,
How do modern chemists characterize the products of chemical reactions?
chemists need to characterize the products of their chemical reactions so that they can be sure of the structure and composition of the molecule they have made. One common way is by measuring the melting point of a solid substance. This doesn't provide specific information about the arrangement of chemical bonds in a molecule, though, so more advanced techniques are required to completely characterize a molecule. These techniques often involve using electromagnetic radiation to probe the energy levels in the molecules. See Physical and Theoretical Chemistry for more on these topics. Knowledge of what energies/wavelengths of light the molecule can absorb can be related directly to structural features of the molecule.
What is a chalcogen?
A group 16 element, oxygen, sulfur, selenium, tellurium, polonium, or livermorium.
What is a phase diagram?
A plot that shows the phase in which a material is expected to exist as a function of variables like temperature and pressure.
Why did William S. Knowles, United States, and Ryojin Wayuri, Japan, win the Nobel Prize for Chemistry?
For their work on chirally catalyzed hydrogenation reactions and K. Barry Sharpless, United States, for his work on chirally catalyzed oxidation reactions. Half of this prize was awarded to Knowles and Noyori for their work with chiral catalysts that chirally catalyzed hydrogenation reactions, or reactions in which two hydrogen atoms are added. This work found applications in pharmaceutical production soon after its discovery. Sharpless was awarded the other half of this prize for developing chiral catalysts to carry out oxidation reactions, which represent another important class of reactions in organic synthesis.
Where do I run into organic chemicals in my life?
The food you eat, the clothes you wear, the gasoline in your car, the plastic bags you may, or may not get at the grocery store. The list could go on forever.
Where are electron transfer reactions important?
Electron transfer reactions are important in many areas. In biological systems, processes like photosynthesis, nitrogen fixation, and aerobic respiration. The process your body uses to make energy using oxygen, all rely heavily on electron transfer reactions. Electron transfer reactions are also frequently used to obtain pure metals from ore. Electrochemical cells, see analytical chemistry, also rely on electron transfer reactions. The batteries that power your cellular phone and other devices use electron transfer reactions to do so.
Where does rubber come from?
Rubber trees. No kidding. Natural rubber is collected from rubber trees, like maple syrup comes from maple trees. Except the syrup is latex sap. It's a polymer of isoprene where each carbon-carbon double bond along the chain has cis stereochemistry. Although there are man-made alternatives produced artificially. Even today about half of the rubber produced each year on our planet does come from rubber trees.
How is gas chromatography different than liquid chromatography?
Gas chromatography involves first vaporizing the sample, while liquid chromatography typically involves making a solution or suspension of the mixture to be separated. In gas chromatography, all of the molecules in the vaporized sample will have the same average kinetic energy, but the different chemical species will each have different average velocities, which are determined by their molecular weight. Heavier molecules will move with slower average velocities than lighter ones. This is how separation of different chemical components is achieved in a gas sample. In liquid samples, the mixture is dissolved in a solution that flows over a stationary phase, which interacts differentially with different chemical species in the sample. These different interactions with the stationary phase cause some compounds to move faster than others through the chromatography column. And this is the basis for separation via liquid chromatography.
What is electrical resistance?
Electrical resistance describes how a material opposes an electric current through the material. The resistance is related to the applied voltage through the relationship R equals V/I, where V is the applied voltage and I is the current through the material. Typically the resistance is a constant, and thus the current will increase linearly with an increase in the applied voltage. This relationship is Ohm's law. As you can see, a material with a higher resistance, R, will have a lower current, I, for a given applied voltage, V.
What is natural gas, and where does it come from?
Natural gas is primarily composed of methane, CH4, and like other hydrocarbons. Methane provides energy via combustion reactions. Natural gas is typically found underground, near sources of petroleum, and can be pumped up via pipelines. Actually, natural gas does not have a smell, so it is mixed with a small amount of a strong-smelling thiol compound so that you can tell if you have a gas leak in your home.
Why did Gerhard Herzberg, Canada, win the Nobel Prize for Chemistry?
For his contributions to the knowledge of electronic structure and geometry of molecules, particularly free radicals. Herzberg was a famous physicist and astrophysicist, and he was awarded the Nobel Prize in Chemistry for his great achievements in molecular spectroscopy. A particular demonstration of his skill in spectroscopy was his investigations into the role of free radicals in chemical reactions. Free radicals had long been a difficult target to study, owing to their relatively short lifetimes, on the order of millionths of a second. Herzberg's talent with spectroscopy allowed him to address this and other similarly challenging, and interesting, problems. Why did Christian B.
Has any legislation been passed regarding the implementation of green chemistry?
Also yes. A couple of examples are the registration, evaluation, authorization, and restriction of chemicals. REACH program in Europe and the California Green Chemistry Initiative in California in the United States. The purpose of the REACH program is to require that companies make data available that demonstrates the safety of their products. This includes the potential chemical hazards during the use of a product. And it also describes means of restricting the use of specific chemicals. A similar piece of legislation, the Toxic Substances Control Act, exists in the United States, but this has received criticism for being far less effective. The California Green Chemistry Initiative was approved in 2008. Requiring the California Department of Toxic Substances Control to place priority on specific chemicals of concern.
What makes mercury so dangerous?
Mercury can be absorbed through the skin, making it particularly dangerous to handle. Organometallic mercury compounds, like dimethylmercury, CH3HgCH3, are particularly dangerous and have caused the deaths of a number of laboratory research chemists. Most research on this most toxic of mercury compounds has ceased. Be careful cleaning up that broken thermometer.
What is a superalloy?
Superalloys are alloys that display a particularly excellent ability to resist deformation under stress at high temperatures, along with good resistance to corrosion and great surface stability. Most often, a superalloy involves nickel, cobalt, or nickel iron as the base alloying element. Superalloys have been used primarily in turbines and in the aerospace industry.
What is an ionic bond?
Attraction between two oppositely charged ions.
What is experimental error?
Experimental error describes the uncertainty or variation in a measurement that is repeated several times. This variation could come from many sources, such as limitations in the quality of the device being used to make the measurement, the ability of the experimenter to accurately repeat the experiment or measurement, or something intrinsic about the quantity being measured.
What is a Lewis base?
A molecular that can donate a pair of electrons.
How did pharmaceutical science get started?
Paracelsus is credited with being the first person to use chemicals in medicine. Before Paracelsus, people believed that illness and disease were caused by an imbalance in the patient. Hippocrates thought it was an imbalance of the four humors: blood, phlegm, black bile, and yellow bile. And Galen furthered these ideas by assigning a symptom to an imbalance of each of Hippocrates' humors. These theories supported the use of medical techniques like bloodletting. Paracelsus believed that illness was the result of something from the outside world attacking inside one's body, and that some of the illnesses could be cured by chemicals. He is also known for proposing the basis of toxicology, namely that dosage was critical to whether a substance was poisonous or not.
What is a flame, really?
From a chemical perspective, a flame is the visible light product of an exothermic oxidation reaction. This reaction takes CH bonds and oxygen, O2, in the atmosphere and starts a radical chain reaction that keeps going as long as there is still fuel and oxygen around. Actually, bonds other than just CH bonds can give rise to burning. That's just what's happening during the burning of objects you're most familiar with, like candles and wood fires.
Does putting urine on a jellyfish sting really help?
You may have heard of this before, but it seems that it may just be an old wives' tale. Washing the area of the sting with salt water is recommended so as to deactivate any of the stinging cells from the jellyfish that may be present on your own skin. Fresh water may actually reactivate the cells that caused the sting, causing you further pain.
What is a positron?
A positron is the antimatter counterpart of the electron. It has the same mass and spin as an electron, but with a charge opposite in sign and equal in magnitude to that of the electron. If an electron and positron collide, they can annihilate each other and release their energy in the form of a photon.
How does blood clotting take place?
Platelets in the blood send a message to constrict blood vessels in the area surrounding a wound. Platelets gather to block the flow of blood coming out. At the same time, a chemical messenger called prothrombin activates an enzyme called thrombin, which then produces a species called fibrin. Fibrin forms threads to block the wound even better.
What is UV/vis spectroscopy?
Electromagnetic radiation in the ultraviolet and visible region of the spectrum. 40 to 1000 Thz, is higher in frequency, and thus energy, than that in either the microwave or infrared. This makes it a good match for the larger energy level spacing associated with transitions between electronic energy levels. UV/vis spectroscopy can be used to study molecules in any phase. However, it is most commonly used for liquid samples.
How was the theory of phlogiston disproved?
Antoine Lavoisier, an 18th-century French chemist, disproved the theory of phlogiston by showing that combustion required a gas, oxygen, and that gas has weight. Lavoisier did this by burning elements in closed containers. These solids gained mass, but the total weight of the containers did not change. What did change was the pressure inside the vessel. When Lavoisier opened the vessel up, air rushed in, and the total weight of the vessel increased. So Beecher had it backward. Oxygen was being used up by the candle instead of phlogiston being given off by the flame.
What is a watt?
A watt is the standard SI unit of power, representing 1 joule of energy per second. It is named after a Scottish engineer by the name of James Watt. To give an idea of how much energy this is, light bulbs in your house typically use about 25 to 100 watts. New types of light bulbs are doing a bit better than this, actually. For comparison, if you are doing manual labor over the course of a long day, your body will average somewhere in the range of 75 watts of power output.
How much CO2 production comes from automobiles?
According to the Environmental Protection Agency, EPA, the transportation industry as a whole accounts for 33% of all CO2 emissions in the U.S. Automobiles used for personal transportation account for 60% of this, or 20% of total U.S. CO2 emissions. The remainder comes from other sources of transportation, such as large diesel vehicles and airplanes burning jet fuel.
Why is diamond so hard?
Pure diamond is a crystalline form of only carbon atoms. The crystal lattice is three-dimensional, and all carbon atoms are attached to 4 other carbon atoms in a perfect tetrahedral geometry. Because the lattice repeats in all 3 dimensions, there is no easy way to distort the structure, making it a very hard material. The structure of graphite, for comparison, is many stacked up layers of carbon atoms. These 2-dimensional sheets can slide relative to one another, making graphite relatively soft in comparison to diamond.
What is a spin?
A type of angular momentum possessed by electrons and some other particles.
What is an atom?
Smallest unit of a chemical element.
Why did Hermann Emil Fischer, Germany, win the Nobel Prize for Chemistry?
For his work on sugar and purine synthesase. Fischer showed that various molecules from plants and animals, which were initially not believed to be related, were in fact structurally similar. He went on to show that various sugars could be made in the lab from simple chemicals like urea, with the ability to make sugar molecules. He proceeded to establish the three-dimensional structure of every known sugar.
What chemicals are used to preserve food?
There are two main types of food preservatives: those that prevent oxidation and those that prevent bacteria or fungi from growing. The first are appropriately named antioxidants, and these molecules work by reacting with oxygen themselves. Unsaturated fats are common targets of oxygen, which causes foods to become rancid. Antioxidants provide an even easier target for oxygen to attack, preventing O2 from wreaking havoc in other ways. Natural antioxidants include molecules like ascorbic acid, vitamin C. And there are also many unnatural antioxidants on the market. The second class of preservatives are those that stop the growth of bacteria and fungi, like mold, from growing. Many of these preservatives are acidic molecules that can be absorbed into the cells of bacteria. If enough acid gets into the cell, basic biochemical functions, specifically fermentation of glucose, slow down enough that the cell dies, and your food stays fresh.
Is everything made of molecules or atoms?
Basically, yes. The only material things that aren't made up of atoms or molecules are the subatomic particles that make up atoms. Anything you find around your house, office, or anywhere else is made of some combination of atoms that are on the periodic table.
What molecular properties do theoretical chemists try to calculate?
Theoretical chemists try to calculate pretty much every molecular property there is. If there's a property we've discussed somewhere in this book, odds are that a theoretical chemist has worked on ways to calculate its value for atoms or molecules.
What is the flash point of a substance?
The flash point of a substance is the temperature above which it can form an ignitable vapor in the air. This requires an ignition source. The vapor may no longer burn if a source of ignition is not present.
Why did Harold Clayton Urey, United States, win the Nobel Prize for Chemistry?
For his discovery of heavy hydrogen. Urey received the Nobel Prize for isolation of deuterium. D2, by distilling a sample of liquid hydrogen multiple times. He is perhaps better known, amazing that he's better known for something other than what he received the Nobel Prize for his work on the Manhattan Project. Working at Columbia, Urey and his team developed a method for enriching uranium by gaseous diffusion. Finally, after World War II ended, at the University of Chicago, Urey and Stanley Miller, one of his graduate students, showed that a mixture of water, ammonia, methane, and hydrogen could produce amino acids by exposing the mixture to electricity. The experiment was designed to simulate conditions in the early days of our planet, and clearly showed that organic molecules that form the basis of all life can be made from basic inorganic building blocks. And a little spark. After Urey's death, it was shown that over 20 different amino acids were present in the mixture. An even more remarkable result than Urey and Miller claimed in their initial publication.
What is the difference between a qualitative and a quantitative observation?
Quantitative observations are, as the name suggests, observations that attempt to quantify how much of something there is. Chemists are often interested in measuring concentrations of chemical species, the amount of energy released in a reaction, the lengths of chemical bonds, and numerical values associated with a multitude of other chemical properties. A few examples of measurements an analytical chemist might make are determining how much fat is in a cookie. About 10 grams, how much CO2 is in the air, about 390 ppm, or how much lead is in your drinking water. Hopefully very little. Qualitative observations are observations that tell about a general property of an object, but don't quantify exactly how much of something there is. Typically this means the observation isn't giving a number of something, but rather it is describing a quality of an object. Examples might be that a piece of candy tastes sweet, that the sky is blue, or that a circle is round. Some observations can be tough to classify as strictly qualitative or strictly quantitative. For example, a judge at the Olympic Games may assign a score of 9.5 to a gymnastics performance, indicating that she really liked it. Quantitatively, this allows for a comparison among the different competitors, but it still feels somewhat qualitative since it reflects a judge's opinion and could be influenced by factors like how carefully the judge observed the performance, what elements of a gymnastics routine the judge values the most, or even just by the judge's mood.
What's in Jell-O, that makes it so jiggly?
Gelatin, which is a convenient and pleasant term for the mixture of proteins and shorter peptides that are obtained from boiling byproducts of the meat and leather industries. Mostly bones and pig skin, makes Jell-O, jiggly. The hydrogen bonds between peptide strands allows gelatin to form a network in the presence of water, which is what the physical structure of the gel is that you are familiar with. These hydrogen bonds can be disrupted with heat, which is why you boil Jell-O, before pouring it into a mold. Once it cools back down, the network reforms in whatever shape the liquid is in.
What is PET, polyethylene terephthalate?
Polyethylene terephthalate is a thermoplastic that is used in both textiles, where it's called polyester, and in bottling, where the same polymer is called PET. PET is an alternating polymer of ethylene and terephthalate monomers. This material is really good at preventing gases from diffusing through it. So it's great for keeping carbonated drinks fizzy.
How large are molecules?
Molecules span a wide range of sizes. The smallest molecules contain only 2 atoms. And these diatomic molecules have length scales that are approximately the sum of the atomic radii of the constituent atoms. The smallest molecule, HH, has a length of only 0.74 angstroms, 7.4 x 10 to 11 m. Larger molecules can be comparatively quite large. Biologically important molecules, like proteins, often contain thousands of atoms. Polymers, which are highly linked networks of covalently bonded atoms, can be even larger still, sometimes becoming so large they are visible to the naked eye.
What is a van der Waals radius?
The radius of an imaginary sphere that can be used to represent the effective size of an atom.
What is interference?
Superposition of 2 or more waves, resulting in either higher constructive, or lower, destructive, amplitude.
What do atomic orbitals look like?
There are 3 main shapes of orbitals relevant to most of chemistry. And these are referred to as s, p, and d orbitals. The designations s, p, and d are abbreviations for sharp, principal, diffuse, and fundamental, which have historical significance describing results of early experiments to probe the electronic structure of atoms. You can see what these orbitals look like in the graphic on the preceding page. The shapes of these orbitals are determined by their orbital angular momentum, which is a property that describes the motion of the electron around a nucleus.
What are green chemical products?
Green chemical products are products that were designed with the principles of green chemistry in mind so that they will not have harmful effects on human or animal health or on other aspects of the environment. Since the advent of green chemistry, a tremendous number of green products have been developed, ranging from safer household paints to greener cleaning products to new types of plastic products. You may have heard the phrase benign by design, which is often used to describe such products.
How do planes stay in the air?
Airplanes are very heavy, so the force required to balance gravity and keep them in the air must be large. The engines propel the airplane forward, but we need to understand what gives the upward push or lift necessary to keep the plane in the air. This lift comes from the shape of the wings, which are typically curved on the top and flat on the bottom. This design requires air to flow more rapidly over the top of the wings than over the bottom, which creates a lower air pressure above the wing than below. The lower air pressure above the wing is what lifts the plane off the ground and keeps it in the air. This is commonly referred to as the Bernoulli principle. If you blow across the top of a sheet of paper, you will see it lift into the air for the same reason.
Can there be more than one solid phase for a given substance?
Yes, solids can adopt different types of microscopic arrangements. If there is a repeating pattern to the atoms in a solid, it is called a crystal. If the ordered structure exists for the entire material, then the phase is known as a single crystal. Think of a diamond. If a sample is a bunch of individual crystals, then we refer to the material as polycrystalline. Lots of solids also just have no pattern to the arrangement of their atoms, and this class is known as amorphous.